Let us move to the next topic, test cross. Before going to learn test cross, let us see Mendel's empirical approach. Mendel's analytical and empirical approach. We all know Mendel chose two different characters, that is contrasting characters. Let us see what are the contrasting characters Mendel chose. These are the characters. There are 7 different characters: height of the plant, plant height, seed shape, cotyledon color, flower color, pod form and position of the flower, flower position. So, these all 7 characters, 1, 2, 3, 4, 5, 6 and 7. All these 7 characters are chosen by Mendel for his hybridization experiments. These all characters are expressed in the chromosome in the form of gene, that is the height of the plant, which is represented in the form of LE, capital L and small e, and the dominant character of plant height is tall and recessive character is dwarf. The dominant character is tall, recessive character is dwarf, which is represented the gene as LE. The seed shape, the gene is noted as R, and the contrasting character is round and wrinkled. Cotyledon color, the gene is represented as I, and the contrasting trait is yellow and green. Then color of the flower, the gene is A. The contrasting character is purple color and white color. Then pod color, which is represented, gene is represented in the form of GP, and the contrasting trait is green and yellow. Then form of the pod form represented the gene as V, and the trait is inflated and constricted. Flower position, axial position and terminal position, the gene is represented as FA and the position is axial and terminal. So, these are the contrasting traits. So, now we are going to learn the Mendel's analytical and empirical approach. So, in all his hybridization experiments, Mendel chose 2 contrasting characters, dominant as well as recessive, 2 traits which consist of which exist 2 different factors. For example, the height of the plant, which represent tall and dwarf, tall and dwarf. Let us recall what we learned in the F1 in the monohybrid cross. Monohybrid cross is a cross of single gene which contains 2 different alleles. So, tall and dwarf, the contrasting trait is tall and dwarf. Under monohybrid cross, this tall, which is represented as capital T and this is small t. So, these are the parent. When you take out the gametes, here capital T and small t. So, here these are the allele, but the gamete is capital T and small t, capital T and small t. So, in the F1 generation, it produces this F1 capital T small t. So, capital T small t is all are heterozygous tall. So, this capital T and small t. So, in F1 generation, these all shows the character tall, heterozygous tall. With these all plants in F1 generation which shows heterozygous, all are tall. But what about this trait? This dwarf character is not disappear, but it is hidden or masked. In this character, capital T as well as small t is there. But this small t will not express in F1 generation, it will be hidden or masked, and it will reappear when at the F2 generation. So, in the F2 generation, which gives the ratio when this F1 is selfed, capital T and capital T, it gives the ratio when it is self, it produces capital T, small t, capital T, small t. So, it gives capital T here, capital T, small t, capital T, small t, small t, small t. So, this is homozygous tall, these two are heterozygous tall, and this is homozygous dwarf. So, it gives the ratio 1:2:1 ratio. Now, this small t is expressed, but in the F1 generation this dwarf character is hidden or masked and it is expressed in the F2 generation. F2 generation this dwarf will be expressed in 1/4 of this. Out of this 1/4 it expressed. Dwarf will be expressed in the F2 generation. So, Mendel's this approach is really an outstanding approach. So, Mendel's analytical approach is truly an outstanding approach, and his experiments are his meticulous experiment, his well planned, careful observation of his meticulous is carefully observed, careful observation of his experiment, meticulous work, and the exceeding the procedures under his experiments, which shows that there is a hereditary unit is present. The hereditary unit, which is now it is represented in the form of gene. So, now the hereditary unit is known as the name gene. The same gene is explained in Mendelian experiments. So, that hereditary unit or the factors, the hereditary unit nowadays it is represented in the form of gene. So, this law or empirical approach, he was the only scientist, Mendel was the only scientist, he explained this type of quantitative analysis in scientific experiments. In breeding plants, he find out this quantitative analysis. So, this governing by this law, which is called as empirical laws. Based on his results, he proposed a law, they are the empirical laws. Now, let us see the test cross. What is meant by test cross? Test cross is a cross of unknown parent is crossed with recessive homozygous recessive dwarf plant, recessive trait. So, here let us take the example, the same monohybrid experiment, monohybrid cross. In monohybrid cross, we are taking the height of the plant. So, this same example, let us see the same example, monohybrid cross. The characters or the traits are tall and dwarf. So, in this 3:1 ratio it gives phenotypically and 1:2:1 it gives genotypically. In the F1 generation, the same experiment, when monohybrid cross, it is taken as capital T and small t, that is tall and dwarf. F1 generation, it produces all plants are tall. All the plants are tall in F1 generation. But F2 generation, here it gives this ratio 1:2:1 ratio. At first, this tall plants, this dwarf is hidden in F1 generation, but it is expressed in F2 generation. Now, this dwarf plant is, so F2 generation, it gives tall as well as dwarf. Now, this F2 dwarf is crossed. Mendel crossed this F2 dwarf, self crossed. It gives dwarf plant in F3 as well as F4 generations. So, F1 is first filial generation, F2 is second filial generation, F3 and F4 third and fourth further generations. When they make a cross between this dwarf into dwarf, it produces homozygous dwarf. So, Mendel concluded that if F2 dwarf is homozygous dwarf is crossed, F3 and F4 generation, it produces the dwarf plant. So, it is homozygous, dwarf is homozygous. But what about this tall plant? Tall will express in the form of either capital T or capital T small t. So, it is homozygous as well as heterozygous. So, how can you predict the tall plant whether it is homozygous or heterozygous? To find out this, there is a unknown parent, unknown character is crossed with F2 dwarf, that is these two crossed with small t, homozygous dwarf. This type of cross is known as test cross. So, test cross is a cross which determines the height of the plant, the tallness, which meant for homozygous or heterozygous. So, what is the definition of test cross? Test cross is a cross between unknown character crossed with a double recessive. That is, here we are taking the example as small t, small t. So, this can be predicted. Let us see how it gives the answer. The first example, this heterozygous tall, capital T small t into small t small t. So, first explanation of test cross is heterozygous tall is crossed with homozygous dwarf. So, this is the parent. It contains capital T and small t. It contains small t and small t. So, the gametes, these are the gametes, capital T, small t and small t, small t. So, it gives capital T small t is to small t, small t, tall and dwarf. So, when you take the ratio, it produces, it gives, the parent is capital T small t. So, capital T small t and small t small t. So, it produces capital T small t, capital T small t, this is small t, capital T small t, here small t, capital T small t. So, it gives tall, heterozygous tall and dwarf. So, in test cross, it gives the ratio, these two are capital T small t, these two are small t small t. So, it gives the ratio 1:1 ratio. 1 heterozygous tall is to 1 homozygous dwarf. If you cross this homozygous capital T tall into homozygous dwarf, any one of the parent with a recessive trait. So, it gives, here what are the gametes, capital T, this is the parent, gametes capital T, here small t, small t. So, it gives capital T, small t, capital T, small t. So, it gives capital T, small t, capital T, small t, all tall plants. So, in this when you make it in the form of checkerboard, it gives here capital T, here small t, small t. So, capital T, small t, capital T, small t, capital T, small t, capital T, small t. So, now all these are tall. All these are tall, heterozygous tall. Everything is heterozygous tall. So, this type of cross is known as test cross. So, in the first cross, we take the heterozygous tall with homozygous recessive. Here, homozygous tall with homozygous recessive. Here, heterozygous tall with homozygous recessive. Here, homozygous tall with homozygous recessive. So, this is the explanation of test cross. Then, what is the molecular explanation of this tall and dwarf? Tall and dwarf. How can we predict this tall and dwarf characters? So, in this tall and dwarf molecular explanation, here find out the molecular explanation of mental. This tall, what is the gene which possess? What's the functioning protein? This tall plants, that is capital T plant, it encodes the gene capital L small e. The gene which is present in this is tall plant is capital L small e, that is LE. So, you just remember this plant height, that is length. So, we can remember the first letter L and E. So, length of the plant. So, LE is the gene present in it. So, which encodes the enzyme which synthesize gibberellin. So, the gibberellin precursor of gibberellin, synthesizing enzyme which converts gibberellin into GA1. So, tall plant which possess LE, the gene which possess here is capital, either it is in the form of capital L small e or capital L small e and small l small e. Whether it is homozygous form or heterozygous form, when LE is present, which encodes the enzyme which is the precursor molecule which converts gibberellin into GA1, the enzyme which activate GA1. So, GA1 is a gibberellin synthesized enzyme which activate the synthesis of gibberellin. This is encodes in tall plants. But in dwarf plants, this encodes the enzyme is not functioning. So, here the functioning enzyme is GA1, it converts, the gibberellin synthesized enzyme is converted into GA1, that is present in tall plant. So, it expresses the tall character. But dwarf plant which do not produce, so it is in recessive form. Look at this, this is the tall trait, this is dwarf. The tall which possess either capital LE homozygous or capital LE small le. Here capital L is present, length LE. So, which express tall, these two traits which express the tallness. And what happens here, gibberellin precursor molecule is present in LE, capital L small e, and this converts this gibberellin precursor molecule is convert into GA1. So, GA1 is the active form of gibberellin. So, it produces, it shows the character height, the tallness of the plant. But this is the dwarf which is recessive trait, recessive character which possesses small l small e. And here the gibberellin precursor molecule, it is inactive form, it is not convert into active form like GA1. So, it is gibberellin. So, here gibberellin is not produced. So, it shows the character dwarf. So, this is the molecular explanation of tall and dwarf. So, once again we review what you learned today. First question, Mendel's analytical and empirical approach, and remember what is monohybrid cross and what is test cross. Two examples of test cross, that is heterozygous as well as homozygous, and these are the character which encodes the gene, and what is the molecular explanation of tall and dwarf. Learn all these questions before the next class. Thank you.